So I guess that we can start with the presentation of David Jeffery. So welcome to everyone to this seminar, and I will introduce Dr. David Jeffery from University of Adelaide. He's associate professor in the Department of Wine Science. And after completing his PhD in synthetic organic chemistry, David began his wine chemistry career as a postdoctoral fellow at the Australian Wine Research Institute. And then he moved to the University of Adelaide in 2010 and is now an associate professor in the department I told before. David is co-author of the book Understanding Wine Chemistry and has published over 110, already 20 peer-reviewed papers. And please, David, I want to acknowledge your availability to impart this seminar in our institute. And I think that's nice for every one of us to hear. It's a pleasure to have you here, and I give you the word, and you can start whenever.
Thank you very much for the invitation, Maria Pilar, and it's very nice for me to be presenting to your colleagues there in the morning for you. So yeah, I'd like to present on some analytical chemistry and how I use this to undertake grape and wine science. So I kind of will cover a little bit of the background that you've already mentioned, just so people know where I've come from. So I didn't do anything to do with wine science for my undergraduate or my PhD. So I did a Bachelor of Technology, kind of focusing mostly on analytical chemistry, but pretty good fundamental chemistry background with organic and physical chemistry. The usual things that undergraduates might do in a chemistry degree. Then I did, in Australia, we have an extra year where you do a research project, an honours year. So I did a synthetic chemistry project there, doing some heterocyclic chemistry and learning skills about NMR. So the usual analytical techniques that synthetic chemists use. So NMR, GCMS, IR, infrared. So those sorts of things that you characterize new molecules with. Then I did a PhD, and this was again in synthetic chemistry. So I did a total synthesis of this molecule that you see on the screen now, which came from this marine sponge, this lovely blue-looking marine creature that you see on the right-hand side. So some biologists isolated UV active compounds from this creature and identified or proposed the structure. And this is often what PhDs involving synthetic chemistry are all about, total synthesis PhDs, where then you try to make this structure that was proposed by the people that isolated the molecule. So that's what they isolated or what they thought on the screen, and I used various synthetic chemistry techniques over the course of my PhD to produce that compound. Actually, as it turned out, the stereochemistry, so the way that those groups are orientated in space in 3 dimensions, was actually incorrect. So not only when doing a total synthesis do you create new synthetic methodology, but you will also verify whether the structure that was reported is actually correct, and in this case it wasn't. So but based on a really good fundamental understanding of chemistry, and particularly organic chemistry and analytical chemistry, I carry those sorts of things through in the research that I do today. In fact, where I first began, as you mentioned, the Australian Wine Research Institute, they hired me not because I knew about wine, but because I knew about chemistry. And I guess they figured that they could teach me what I needed to know about wine. And certainly that's the case. Over 5.5 years or so, I learned a lot about wine chemistry. And I think it was easier for me to gain that understanding by having that really strong fundamental chemistry background. So I started there as a postdoc and worked for a couple of years, and then worked my way up to team leader of the flavour group. And perhaps you or some of your colleagues might recall a researcher by the name of Mark Sefton, a very well-known flavour researcher at the Australian Wine Research Institute for like over 20 years or so. He was there. So when he retired, I took over his leadership of the flavour research team at AWRI. Then a position came up at the University of Adelaide, where I moved to in 2010, and then I've worked my way up to associate professor now, you know, doing the usual things that you do within a university in terms of research and teaching and service to the university and to the profession. Along the way, I've gained a few things. So I did a graduate diploma in online learning, so a tertiary teaching qualification. I obtained a couple of awards there. So that one, the Peter Alexander Medal, was for excellence in Australia in analytical chemistry for the first 10 years of my career. And that was analytical chemistry related to wine. So, you know, that's kind of interesting that a chemical institute in Australia thought that, you know, the analytical chemistry that I applied to wine was worthy of winning their medal. And you mentioned the textbook there, and there's a picture of it, and I guess people may be a bit familiar with that. And you can see in the background a lovely big periodic table on the wall in my dining room. This chemistry textbook received a special mention from the OIV under books in enology in that year that we submitted this, so that's kind of cool. And we're actually working on the revised version of this right now, me and Andy Waterhouse and Gavin Sachs. Unfortunately, we're not doing a Spanish edition of it yet. It's published in Chinese, but the next edition of this will also be in English. But I presume the fact that everyone's listening to me and following and that they'll be able to make sense of that book written in English anyway, so it's not a big deal. So what about wine then? And why is it important and you know, relevant to Australia? So it's a multi-billion-dollar contributor to Australia's economy, just like it is in Spain and Italy and France, and you know, the big wine-producing nations. I think it's quite impressive that Australia, with a population of, you know, 26 million people, so not a very large population, it's a huge country, but that we're the sixth largest wine producer globally and fifth largest exporter, or fourth by value. And you can guess who's above us, and that's Spain and Italy and France. It creates many thousands of jobs. Here's some other data in terms of the tonnes crushed. So 2.03 million. I'm not sure what the total annual production is in Spain, but you know, this isn't you know, it's not the largest production in the world, but around the grapes themselves, around $1.56 billion Australian in value, and around 700 Australian dollars per tonne on average for the price. You can see here the kinds of yields that have been produced over the last decade or so. So in fact, in 2021, that was a record year in Australia, where we produced over 2 million tonnes of grapes. Now, in terms of the states of Australia, so that's the country there that you can see. Let me see if I can get so I'm in South Australia here, which produces around half of Australia's wine production. You can see other states, so New South Wales, where Sydney is, the capital city, Sydney, and in Victoria, a little bit less, and yeah, South Australia. So places like the Barossa Valley, McLaren Vale, some of those names you might be familiar with. So I work in the state that produces over half of Australia's grapes for wine production. A little bit more in terms of the distribution between the colours of the grape, so a little bit higher production of red grape varieties for winemaking than white. And over here in particular, you can see the different varietals. So you might know, or at least I'll tell you, that Shiraz is a very popularly produced red grape variety. Second in Australia in production terms is Chardonnay, and then Cabernet, and then Merlot and Sauvignon Blanc, which is kind of interesting for another white varietal, that Sauvignon Blanc is there in 5th place. And you can see some of the values here for the tons produced, or per ton. Shiraz fetching nearly 900 Australian dollars per ton. Cabernet Sauvignon, a globally important variety, almost $800 per ton. So these are some of the relevant figures that kind of make it important to undertake wine research in Australia, because this is what's actually really put us on the map, I think, in terms of understanding wine production from the vineyard all the way through to consumers, but particularly wine chemistry and microbiology and stability. These are the things that have really underpinned Australia's success, I think. So why is there success? Well, I guess analysis is a key to that, and I think you probably all appreciate this. So you really can't manage things that you can't measure, and this is true whether it's wine chemistry or in business or any other field, really. So the fact that we have an ability to measure things is what's been very important, and even as simple as pH control. This is really where things kind of started. So back in the 1930s, a chemist at a wine company said we should buy a pH meter. And there was some resistance, and this is going to be a very expensive piece of equipment or whatever. But then just purely understanding the effect of pH on controlling spoilage microbes, understanding the effects of acidity. And I'm not sure if you realise, but Australia being mostly warm viticultural regions, we are allowed to add acid but not sugar, so we don't have problems with fruit getting to sugar ripeness. But then we have lower acid concentrations, so we can add tartaric acid during winemaking, whereas some other cool climate countries perhaps add sugar and not acid. So there's a little bit of a difference there. But in terms of where the Australian industry started, or its success started, was way back in the 1930s with just measuring pH and going from there. Because the other thing is, a lot of Australia's wine was exported to the United Kingdom at that stage. So spoilage during transport or transit was a very big thing. So getting on top of that then really underpinned our ability to produce high quality table wines that we could then ship around the world. Of course, there's many things to consider in wine. The composition of wine is very complex. So having the ability to measure and different techniques to measure things in wine is very important from a chemistry point of view. And a lot of you, I'm sure, would be familiar with the different analytical techniques that are available and that we need to apply to understand the range of constituents that can be found in grape and wine. This is what excites me about it, the fact that it's a natural product, a very complex one, but it's ever changing. It evolves over time. So when you look at a wine today, it's not necessarily the same as it would appear either by sensory or even analytically. The chemistry is not the same after a few years, and it continues to change, which makes it really interesting but also a challenge to understand. Of course, we don't just measure things for the sake of being able to measure them. They need to be important. So important to the sensory properties of wine, to the quality or style of wine, especially things around stability, because in the end, all of this is geared towards what consumers like. You need to be able to sell the wine. It doesn't matter how nicely it's made or how beautiful the story is or whatever. If you can't sell the wine to people, then it's not really much good. I mean, companies and producers need to be able to obtain an income from the products, and the better the products are, the more likely they can obtain a higher income. This is obviously, you would be all aware of this sort of thing, but where the inputs are from. So this is part of the analytical challenge. We've got the grapes, we've got microorganisms involved in winemaking. There's conditions of fermentation and different fermenters or whatever. There's aging and maturation, whether it's in oak or stainless steel, and then bottle aging and under different closures. In Australia, we hardly use any cork at all. We're mostly under screw cap in Australia, which produces its own challenges because of quite a reductive environment for our bottles. You know, not as much oxygen transfer through a screw cap closure as there is through cork. But then it eliminates the variability of cork, although it's getting much better. And of course, it gets rid of things like cork taint. That's not a problem, but then, you know, potentially reductive issues become a problem under screw cap. Other things that we can face is from the environment. So this is a bushfire or a wildfire here, and you might have started hearing, certainly in Australia, it's happened for a long time. In terms of impacting on vineyards. In the US, in California and Washington State, it's started to happen more often. South Africa, but even France now is having wildfires that are potentially impacting their vineyards. So the smoke, the volatiles from the smoke drift over the grapes and land on the grapes, and once they get absorbed inside, and we're not exactly sure of the mechanism still how this occurs, but then those volatile phenols like guaiacol and eugenol and cresols, the usual things that you might expect from charred oak barrel, for instance, plus others, they get glycosylated in the grape berry, so become non-volatile, and then they're re-released either during winemaking or storage or even in your mouth. So when you taste wine, glycosides that are bound can release the volatiles, and you get this perception of smoke and ash and campfire, which is not very nice. And this is something I've worked on in the past, but of course, and not so much anymore. One of my colleagues is a world expert in this area, but this is just to highlight that there's new challenges coming along every day, perhaps, or every year, where we need to do even more research to understand this new challenge to the industry, and that's where analysis and analytical chemistry is obviously very helpful. So I'm going to step through a couple of things that we've been working on. One big area that I've worked on for quite a few years now is varietal thiols. So understanding them, how to analyze them is a big challenge, and then what are the influences of various aspects of winemaking and that. So varietal thiols, polyfunctional thiols as they're also known, they contribute to their character impact compounds. So they underpin the characters of certain wine varieties. They're in a lot of different wines, but certain wines, they're really expressive, like Sauvignon Blanc, for instance. So it's aromas of things like box tree or black currant. So this is something called 4-MSP. And I'd like you to look at the thresholds, the odor detection thresholds. So not only the concentration of these, but then I'll show you a table with odor detection thresholds. These are some of the most powerful in any food or beverage product. But also at this low concentration, low nanograms per litre, that presents an analytical challenge. So there's 4-MSP, there's 3-sulfonyl hexanonol or mercaptohexanol you might know of, the hexyl acetate there. So there's the concentration ranges. The other thing is that we rarely, even as wine chemists, we rarely think about stereochemistry and the fact that if a compound has a chiral center, then there's the possibility of forming enantiomers. And these might possess slightly different thresholds, as we see here for 3SH, but also potentially different aromas, so different sensory qualities as well. And we can see that here with the enantiomers of 3SH, and 3SH gets acetylated by yeast to produce the acetate, and therefore there's also enantiomers of 3SHA. And again, we see that they have slightly different aroma thresholds, and they can be in different concentrations as well, that they're not necessarily equally distributed between these enantiomers. So that's going to influence wine aroma. So the chemistry, the molecules within the wine obviously dictate the sensory properties of the wine, and that comes down to even chiral molecules, especially impact odorants like these, that have slightly different thresholds and different aroma qualities. If there's more of this one than this one, then that can have a big impact on the sensory qualities of the wine overall. So kind of bringing my background together here with the analysis but also the synthesis, we have the capability of synthesizing compounds, and although I could do complex syntheses, we tend not to. We try to do easy ones because my students that work on these projects, they're not synthetic chemists like I was. So they can't do, or I wouldn't expect them to do, multi-step syntheses. But some of these syntheses are reasonably straightforward, and they can achieve them in a few steps. And so things like making deuterated compounds, because our analytical work, this is more or less the gold standard in analytical work, this stable isotope dilution assay or CIDR idea, where you use deuterated internal standards to develop your calibrations, and then the benefit of deuterated standards that are exactly the same as the natural analyte but only contain deuterium, that contain some level of deuteriums rather than hydrogen. They behave exactly the same, except we can tell them apart by mass spectrometry. So if we're preparing a sample and we use this deuterated standard to analyze 4-MSP, and if some 4-MSP gets bound to the inlet of the GC, then some of the standard will get bound. Or if the 4-MSP in the solution that you're preparing somehow breaks down or reacts, then some of the standard would react as well. So we made D10 4-MSP from D6 acetone. You need to start with something commercially available, so D6 acetone or D10 butanol. So just briefly, you're probably not interested so much in the synthetic chemistry, but just to highlight some of the steps that are undertaken, and also the positions of the deuteriums as well. It's important to understand the chemistry. So, I mean, this is how this is made. So starting off D6 acetone, basically a condensation of that to get mesotyl oxide as an intermediate, adding the sulfur group, and then a reduction to free that up. But it's important then to understand where are these deuteriums placed, because next to a carbonyl like this, they're exchangeable. So it's really only these ones here. So D6 4-MSP is the most stable version, because in an aqueous environment, and depending how acidic it is, these deuteriums here can exchange back to protons, so you lose the label. This deuterium here can also exchange back, so you can lose the label. We did the same sort of thing, or similar sort of chemistry to make D8 3SH. There's not so much of a problem in terms of losing the label. We don't have label next to the hydroxyl in this, but even if we did, then it's not going to be lost because that's not in an enolizable position. So anything next to a carbonyl, like we see here, can be exchanged back to the hydrogens. So this is something to keep in mind if you ever look at papers or think about deuterium labeling. If they've only got the labels next to something where the labels can exchange back to hydrogen, that's not a very good standard. The standards really need to have positions that can't be exchanged, and then they'll stay there. Because if you use this deuterated internal standard and you put it in your wine and you leave it for a while, or there's something around about the pH or whatever, where you lose these labels, then suddenly you don't have that marker that you can look for with mass spectrometry, the different mass of this compound compared to the parent. So I've seen mistakes in papers where they even have the deuterium on the oxygen here. And that's just crazy, because that's the first thing they'll exchange, as soon as that goes in an aqueous environment. And I think they've just drawn it wrong anyway, or don't understand where the label has gone. But drawing a deuterium on an oxygen like that would not be a useful internal standard whatsoever. So having an understanding of the chemistry and then being able to synthesize these sorts of compounds has been extremely important for our work. We've made other standards as well, so you know we can take this one and then acetylate it to form 3SHA and that. So just a bit more manipulation there, but basically many of these steps are, you know, not that complicated. That a wine chemist could, you know, basically undertake these as well. But although even still, we have synthesized standards for other groups internationally, I guess because we're set up for it and able to do that. So let me talk a bit about analysis of thiols, because we've done quite a bit of work on this, as I've said. So thiols in general are difficult to analyze. They're very reactive. They're at very low concentrations. So there's methods out there, and very good methods, you know, out of Vicente's lab at Zaragoza, and I'm sure some of you have probably seen these, use these yourself. Okay, so things like doing GC on either from mercury benzoate for the extraction, so that the mercury is very selective for thiols, and then they can be released, or derivatization with something like pentafluorobenzyl bromide to make the thiol less reactive, but also make the method more sensitive, particularly if you're talking about something like negative chemical ionization, where having lots of fluorines in your derivatized molecule really loves to take on a negative charge. So we started looking at those, and you know, here's a bit of a photograph of a setup. You know, but these are complex methods in terms of the preparation of the samples. There's a lot of mucking around. So, you know, we take, and this is a method we developed based on PFBBR, but using a electron ionization benchtop GC, so not chemical ionization. So we needed a lot more wine. It's less sensitive, but you know, measuring out the wine, spiking in the reagents, extracting the wines, doing all this stuff. There's a lot of involved techniques with that, and I thought there's got to be a better way. This is crazy doing this, you know. Takes a lot of time. We could get very good limits of quantification, 40 nanograms per litre below the threshold, but can't do many samples in a day. You know, if you're talking about calibration samples and actual samples, we couldn't do very many at all. So about 25. So I thought, you know, there's got to be a better way than this. And what I really wanted to do was to somehow maybe derivatize thiols and be able to analyze them directly without any pre-preparation or isolation or whatever. That's a bit of a dream still. That's not possible at the moment, except perhaps for very high levels of 3MH. But what we did do was develop a method using HPLC with tandem mass spectrometry, and we use this derivatization agent, which is specific for thiols, so 4,4' dithiodipyridine. But the biggest advantage, I think, of this, other than we're a bit crazy going to HPLC for a volatile compound, but other people are doing that too, but this can be done at wine pH. So we looked at other things like Elman's reagent, DTNB, you know, other thiol specific reagents, but they require pH adjustment of the wine. And I'm not sure if you ever tried that, but to try to adjust a wine to pH 7 or 8 is not very easy at all. And trying to do that on every individual wine is like really time consuming and almost impossible. So we use this. So we take, it's the thiol, it reacts with this reagent. It's basically a thiol disulfide exchange. We form a new disulfide, a mixed disulfide with our thiol, plus this thiopyridine part. And then this is made, actually, this reagent is used in UV visible spectroscopy, where you actually analyze for this 4-thiopyridine that comes off. We didn't care about that. And I mean, you see, you turn a thiol with no chromophore into something that is now UV active. But we didn't care about that, even though I've shown you the wavelengths that these absorb at. We cared because now we've derivatized our thiol, we've put on a nitropyridyl group, which under acidic conditions, this will be protonated, so very good for electrospray ionization that we use with liquid chromatography. So this is the procedure, and this gives us plenty of sensitivity. We can do it with less wine, but 20 mils of wine, spike with labeled internal standard that I told you about before, deuterated standards, adding EDTA to help prevent oxidation. Acetaldehyde is important to mop up to bind any free SO2 so it doesn't interfere with our reagent. Adding the derivatizing reagent, letting this incubate for half an hour or so. We put it through SPE, solid phase extraction, to obtain extracts only for a concentration factor, a little bit of a cleanup, but about 100-fold concentration factor. And then we use LCMS with multiple reaction monitoring. So as I kind of mentioned on the slide before, it wasn't so much that we added a chromophore by adding that aromatic ring. It was more about adding an ionizable group. For electrospray. So then we took that and developed a method for what we consider the 5 most important thiols. So Vicente has done a method on this, so we kind of took or went based on the thiols that were analysed in that. A very good method, but as I mentioned, you know, a bit more complicated or complex to do. Then I think taking a liquid, and this is what drew me to it. We're working with wine, it's a liquid. So if we could keep it in the liquid phase, that seemed to be a more sensible way to go for me. So we took these, you know, what we consider most important thiols. So a couple of those, or 3 varietal thiols that I've mentioned before, and then a couple of others. So something that comes, we're still not 100% sure where benzyl mercaptan comes from, but it's related with aging and sort of smoky characteristics in aged champagne and that. And then furfuryl thiol, which is related to oak wood contact. Again, really low thresholds though, really important or impactful odorants. We had to make deuterated standards for these. So again, with some reasonably straightforward chemistry, we started with the D4 commercial compound and did a reduction, and then added the sulfur group. And similarly here, taking this pentadeutylated benzyl chloride here, adding the sulfur group, and then basically deprotecting or freeing that up. So we made a couple of new internal standards for that procedure. And then, as you do with analytical methods, you need to validate them and check that they work okay, and they did. It worked fantastically. We wanted detection limits around the threshold, the aroma detection threshold of the analyte. So then if there's small changes in winemaking or storage, or, you know, very small changes at the kind of low nanogram per litre level, we'd be able to pick that up. And that's important then, because as I'll kind of talk about on the way, or finish off especially, it's not just the ability to analyze things. You need to be able to do something with that information, help winemakers or understand a problem better, not just purely for the sake of analysis. So we did some infusion mass spec experiments and determined the fragmentation pattern of these things. And here I've shown the acetate 3-MHA and its deuterated version. So this highlights where the deuterium, adding deuteriums in, really helps. These things almost co-elute by chromatography, although the more deuterium atoms you add, the earlier that compound elutes, either by GC or LC. But having deuterium atoms, it acts exactly like the unlabeled or the natural compound that's in wine, but we can tell it apart by mass spec, and that's what we see here. These things fragment to give this portion of the molecule, so really intense 144, which comes from the derivatized part, and a much smaller 111 or a 112 usually. This one's interesting. I'll show you some other ones. No, sorry, I don't have them, but other ones, like for 3SH, without this acetate, they're much simpler. There's the protonated molecular ion, there's a 144 and a 111. So they all give this diagnostic 144 fragment, and we use that in our multiple reaction monitoring pairs. And these are the kind of chromatograms that we get in the end. So very clean, using MRM. So these are the analytes at roughly mid concentration ranges. These are the transitions for the different analytes that we're working on, just shown here. Of course, we record other ones as well. So this is the ones we quantify on, but we record ones that also verify that this is the right peak that we should be looking at, for instance, or this one. But we also have the internal standard in there, which helps us find the peak that we should be looking for, because it effectively elutes at very much the same time. So, you know, mid concentration levels in wine, a few hundred nanograms per litre, or a few thousand in the case of 3MH, or even, you know, tens of nanograms for some of these lower concentration ones. So very clean chromatography once you use multiple reaction monitoring. You know, not easy to see that this is our analyte peak, and not much other noise and that, so gives us very nice limits of detection and quantification. If you want to read more about this, this is the paper that this work was done in. So now we've taken that, I mentioned earlier that compounds are chiral. So there's a few thiols that are chiral. So we took this method and adapted it to chiral analysis. So the same derivatization procedure, but we needed a chiral column, and we didn't know which one to buy. They're not all going to necessarily give us resolution of the enantiomers. And if you've ever tried to buy a HPLC column, a chiral phase HPLC column, they're like, at least Australian dollars, they're like $2,000, like twice as expensive as a chiral GC column. So we couldn't just buy columns and test them ourselves. So what we did, we used it's part of Phenomenex, it's called Phenologics, and you can send compounds there and they'll do screening of the right chiral stationary phase. But we had to then prepare an authentic standard to send to them. So we used a bit of chemistry again, of our derivatives, to be able to send them enough. Like, we're not talking about nanograms per liter of stuff. They were doing UV detection. So luckily we have a method that that puts a chromophore onto our thiol, but they need much higher concentrations to do their screening, because they use UV, not mass spec. So anyway, we prepared a mixed disulfide using some known chemistry. My student did this, and then we produced plenty of this to be able to send for this chiral column screening service. So they did some screening, and they screened a few columns and showed us the results, and these were a couple of the phases that they chose. So we chose one of these and bought it. We didn't quite get the resolution that they did with their system. In fact, they were trialing it on a 4.6 millimeter column, and we use a 2.1 for our mass spec. So fortunately they screened a few, and they actually sent us another column that worked better. We then developed our method, figured out the enantiomer elution order, because we were able to, in the past at least, had synthesized the individual enantiomers of these compounds. So that was important to know which one is the R and which one is the S. Anyway, we developed our method and we can see this is overlaid the internal standards plus the analyte. So we can see 3SH here. It's amazing, this separation of enantiomers is very unusual. So the column that we've chose, or that they thought was best, this amylose 2, these were barely resolved. So this R enantiomer of 3SH came out much earlier. So this is a long method. We'd prefer things to be like this, you know, a very short method. So it's a bit longer than we'd like, but at least we get resolution of the enantiomers. But a really peculiar effect that one enantiomer is retained so much longer or more than the other one. Anyway, we developed and validated the method, multiple reaction monitoring. Here's the spectra that we'd get from 3SH. I mentioned earlier, very simple usually, just the 144 and the 111 plus the protonated parent molecule. Here's the one for SHA, very much before. We chose different pairs to monitor now for multiple reaction monitoring. You can see those here. And now we have a nice chiral method for thiol enantiomers that we can apply and understand various things like winemaking treatments, or how do these relate to the precursors. So we applied this to commercial wines. Now we can get an idea of the distribution of enantiomers. Tominaga, a name you might know of, did this decades ago, but on a chiral GC column. But no one has touched this work since 20 years ago or so, basically hasn't done chiral analysis of thiols. So we analysed a range of wines. You can see the concentrations and of the different enantiomers here. We found roughly 50-50, or what's called a racemic mixture of 3SH, but interestingly the acetate was a 60-40 or sometimes 70-30, so botrytized wine, for instance, it even increases to 70-30. Now this is exactly what Tominaga kind of showed 20 years ago with his GC method, but this I think is much simpler and we can screen a lot more wines, but we can also then start to understand the effects of winemaking and also the formation pathways. And I'll talk about thiol precursors in a second, but now we can understand or relate stereochemistry of precursors to the stereochemistry of the thiols that are released during fermentation. And I think this is really important. I mentioned that thing about botrytized wine. So there's obviously a biological effect as well. So things are where enzymes are involved, they typically are stereoselective, typically giving more of one enantiomer than another. So this is what starts to help us understand what might be an enzymatic effect, what might be a chemical effect. But we've then used this method along with a method that we've got for precursors. So the precursors, the non-volatile grape derived precursors to thiols. I mean, this is still a work in progress, but it's generally thought the main ones are conjugates of glutathione and cysteine, and there's some intermediates, and they are diastereomers. They have stereochemistry too. Glutathione is chiral, cysteine is chiral. So then when you add that to the chiral center of the thiol, you get diastereomers, and we have a method to analyze those in juice and wine. And now we can start to do things like linking the stereochemistry of precursors in grapes to thiols in wine. And I think that's a really cool thing to be able to do. So now thinking about chirality of the thiols themselves once they're released, and the fate. And this is where chemistry, my chemistry background got me thinking, and then understanding wine chemistry as well got me thinking about what are the other possibilities of where thiols can be consumed during winemaking or storage. So we know they can oxidize to disulfides, they can act as nucleophiles. So if phenolics oxidize and form quinones, then they can bind thiols. So all of this means that it's having an effect on wine aroma. If we're losing thiols from solution, then we're losing wine aroma as well. And I think I need to speed up a little bit because I don't want to keep you all day. So we know that acetaldehyde is present in wine, so we got to thinking about what if thiols react with acetaldehyde, acid catalyzed reaction, forming these new compounds, at least for wine, known as oxothiones. They're found in passion fruit, and passion fruit has thiol and aldehyde, so it kind of made sense. So we wanted to develop a method to understand how do thiols react and form oxothiones. Interestingly, I won't go into it, but they're only present, or at least not in great detail. Only the cis isomer we have figured out is present. So this is how it relates to the thiol enantiomers, the R and the S. Then you can get cis and trans isomers of the oxothione. Only the cis ones are present. And this stereochemistry here relates to the stereochemistry of the thiol. So we wanted to now analyze these enantiomers. So we've moved back to GC now for those ones. They're not easily derivatizable, or the thiol is now bound up in this oxothione ring, so we can't do what we did for thiols to analyze them. So back to GC, we had to make a labeled internal standard. We compared, now we had to identify these things in wine, so comparison of mass spectra, retention indices, different GC phases, you know, the usual thing that you do when you're identifying a new compound in wine to make sure you've got it right. Looking at it in different matrices, prepared an internal standard, and then quantified different wines. So as we can see, we didn't do a huge range of wines in this first study, but Sauvignon Blanc, as it's very full or very abundant in thiol, so it makes sense that it has oxythione as well. Chardonnay is interesting, a few other ones. And then in terms of origin, it was interesting to see that New Zealand wines, which are very abundant in, some of them at least, very abundant in thiols, also had the highest concentrations of this oxythione. We also determined the odor detection threshold. Now, if you recall, thiol odor detection thresholds are like, you know, low nanogram per litre. This is 1,000 times higher. So it's not like this is going to be important perhaps to wine sensory qualities, but if it's consuming, you know, hundreds of nanograms per litre of 3SH, then that may well have an impact on a wine aroma. So we've done much more with this, looking at stability of these new compounds, thinking about pH effects and whatever, if they're being stored. So if I just step back a little bit, if these things can form, they can also break down and maybe revert back to the thiol. So we thought they could be a pool that releases thiol over time, perhaps. So we did some studies that looked at different, the ability of these things to, or how stable they were. We also did studies around the evolution of these during fermentation and aging. If they form from acetaldehyde, then perhaps yeasts that produce more acetaldehyde would form more of these. So we looked at studies like with a couple of different yeast strains. So the normal yeast strain that might be used for Sauvignon Blanc, and then what we thought, this is a sherry yeast strain or a floor yeast, which should produce lots of acetaldehyde. It wasn't the case in our study, but here's the acetaldehyde production over time. There's the end of alcoholic fermentation here. So producing acetaldehyde early. We did a co-inoculation experiment as well. Interestingly, this floor yeast didn't produce as much acetaldehyde as Vin13 did. We looked at the evolution of the thiols as well, and you know, you can see that profile here. Here's the evolution of that oxythione. It very much looks similar to when acetaldehyde is being produced, not really when the abundance of the thiol is being produced. Then we did, you know, looked at correlations and that there was a strong correlation in each of these fermentations between acetaldehyde, a negative one here with 3SH, and, you know, reasonably strong correlations with 3SHA in terms of the production of this. So there's various aspects there that to consider that I don't really have time to go into now. But it was found acetaldehyde, interestingly, even though there's tens of milligrams per litre of that versus, you know, thousands of nanograms per litre of this, acetaldehyde seemed to be a limiting factor. But it is an equilibrium between acetaldehyde and the thiol, so maybe that's not unusual. We developed a chiral method to analyse those oxythiones now, found strong correlations between the enantiomers of the thiol, and during this work we actually identified a new oxythione. So from now something called MSPOH, so the alcohol version of MSP. Now that has enantiomers again. MSP is achiral, it doesn't have enantiomers. But when the carbonyl is reduced to a alcohol, then there are enantiomers. So we started looking for this other one now that's arisen from this alcohol here, MSPOH. And you'll notice this interesting structure. There's an alcohol, then 1, 2, 3 carbons away, there's a thiol. Alcohol, then 1, 2, 3 carbons away, there's a thiol. So there's something unique or interesting about this chemistry that it's set up to form these kinds of oxythiones. So we discovered this new one here, only one enantiomer, but it's kind of interesting. We have to prepare authentic standards and labeled compound again. We determined the odor detection threshold of this as well. And what it seems to be is quite a general reaction between 1,3-sulfonyl alkanols, that specific arrangement that I've indicated here and here, that's going to form these new oxythiones, at least in wine. These things, this is brand new, it hasn't been shown in any other food product or fruit or anything. These ones are known in passion fruit juice at least. I'll switch over now to something else because I work on many different things and authenticity is one. So and it's obviously an issue for Rioja or anywhere there's DOC or designated origin wines in particular, because they're the most expensive ones usually. So, you know, annually on a global scale, this is a multi-billion-dollar issue, the issue of wine fraud, and costs Australia quite a few $100 million a year. There's something as simple as putting a fake label on a shonky wine. So Penfolds are very or Grange, I should say, a very well-known Australian high quality or super premium wine. Even just as something simple as putting on this stupid label and trying to sell that as, you know, this high quality wine. There was actually a movie made, you might have seen it a few years ago, called Sour Grapes. It was the first person in the US ever convicted of wine fraud. So there's different options. Of course, there's data, you know, how many grapes are produced in your region. You can't produce more wine than you have grapes, so that's an obvious one. But other than that, you know, there's other ways. But I'm a chemist, so I wanted to do, you know, not packaging. Like there's different things around smart packaging and blockchain and traceability and that, but I wanted to do something that's chemical based. That's much harder to defraud or change. You know, to make to produce or recreate the chemistry, the composition of a wine is much harder, if not impossible, compared to creating a fake label like this. So we investigated a method using this spectrophlorometry, basically. So this technique called A-Team. So there's a specific instrument by Hariba, and they've come up with this technique, absorbance, transmittance, excitation, emission matrix, using this Aqualog spectrophotometer. So what it does is produces or creates data that then, so this is fluorescence, these are the excitation emission spectra. These are some different wines here. You might go, well, that doesn't look very different at all, but you know, if you look closely, there's a bit of difference. But then we use chemometrics and particularly machine learning to look at the patterns of these and decide where have these wines come from. So it's very simple, rapid, doesn't take much wine. There's a bit of pre-processing of that data. Then we did applied this method, you know, to authentication of wines from Cabernet Sauvignon wines from different regions. It's very simple, and I like simple things, as you might have seen with the method for thiols, but take your wine, dilute it with acidified ethanol, sonicate, stir, and then you can analyze it. There's some data processing to be done, so PLS compression of the data and then machine learning modeling. So here's some of the results here from the first work that we did on those Cabernet wines. So Bordeaux, Coonawarra, Margaret River, Yarra Valley. These are regions in Australia versus Bordeaux in France. We used a couple of different chemometrical machine learning techniques, support vector machine versus this one called extreme gradient boosting discriminant analysis. And you can see that worked really well, actually better than support vector machine. And there's other ones. So this is a tree-based algorithm, and it worked really well for multi-class and unbalanced groups. And we keep using that now. So we can classify by region or variety. We can actually fuse data sets. So I brushed past it before, but this Aqualog also collects the usual absorbance and CLab data. So you can fuse those data sets together, and we can actually predict phenolic compound concentrations by doing PLS modeling against HPLC data, and I'll show you that in a second. So we did a lot more wines, many different regions, and you know, you don't get much better than this. People have used MIR and NIR to classify and all other kinds of things, and still nothing gives as good or better results than this. And our method is so simple that you know, I really enjoy it, and I think it's a great method. So you know, fabulous ability to predict the origin, the region, So I'll skip through. This is what I talked about in terms of now we can quantify phenolics with this instrument. So, basically becoming a rapid method to quantify phenolics. Then you can see these different regions, how the different phenolics group those regions to certain extents related to the weather or irrigation, how hot those regions are. We're doing many more things now with this instrument as well, looking at heterogeneity of grapes. This is amazing, right? We're trying to predict wine sensory properties from grape data using this instrument. We can predict the percentage of wine in a blend using this instrument. So I think there's, many more things to be done and that we're going to try to do as well. So my animation's gone a bit funny here, but anyway, in summary, I'll just pop them all up. Actually, it's the chemistry that I have a background in. Organic and analytical is the fundamental aspects of my research. The research, while it's fundamental in terms of knowledge, scientific knowledge, it's also very important in terms of the contribution that it makes to knowledge generated for the industry, a very important Australian industry. A lot of projects that I do are related to aroma volatiles and particularly thiols and trying to understand their formation and fate. These are typically used the usual instrumentation, but then it's how it's applied and in what ways do you use it afterwards. So that's what I mentioned here. They're the usual tools that we all use, and stable isotope dilution assay gives us really a reliable quantification, and having the ability to produce labeled standards is really useful for that. We also make authentic standards as well. You can't just buy thiol precursors, the glutathione or cysteine conjugates, for instance. So it's important to be able to synthesize things for our work. I mentioned around the rapid spectroscopic methods that we're starting to work on. So things around authentication, but then we can do regression with this, predicting concentrations of phenolics that would take hours of HPLC analysis. Once those PLS models are built with HPLC as the reference method, then we can really rapidly predict phenolic concentrations. Ultimately filling in gaps in knowledge through chemical analysis, but cross-disciplinary as well. So understanding from the grape growing and viticultural management techniques, winemaking, production aspects through to sensory. I mentioned this thing about, you can't manage what you can't measure. So we measure things and then we give guidance or can provide information about how those things can be managed. And I sort of touched on this earlier, that the methods we develop are really just the tools. They're not the end of the story or the end of the road for us. Once we have them, that's when the work really starts. It's how we apply them to gain fundamental knowledge or useful information for producers. So that brings me to the end. I'd like to acknowledge, sorry if I went a little bit over time, Maria Pilar, what you were expecting. But so these are some of the people that have worked with me. So Dimitra Capone, I worked with her at the Australian Wine Research Institute. And now I've been very lucky in a project that we've had recently that I was able to employ her myself here. Ruchira Ranawira has done a lot of that aqualog work. Xing Chen Wang and Liang Chen have worked on thiols. Xing Chen is still here with me. Liang is now at ISVV in Bordeaux. Ross Sanders with me at the moment doing some work on some other stuff as well. We collaborate with different places. So this is an ARC training centre that I'm working in at the moment. But also I still work with people at AWRI and an organisation here called CSIRO. We have co-supervisors of a student from Montpellier Subagro, worked with Heriba Instruments for that aqualog instrument there, and our grape growers and winemakers, probably much as they do there for you, really support our work by providing samples or access to vineyards and let us do viticultural treatments or whatever. So that's really, really important. Thank you very much, Maria Pilar, for the invitation, and to your colleagues there at ICVV for listening. So that's it. Thank you. I'll stop sharing now.